Okay, hi everyone. We're starting a new chapter on viruses. I just wanted to mention regarding this first slide, um, this is a dose, a vial of Gardasil, a vaccine which protects against the human papillomavirus. The human papillomavirus causes cervical cancer, which is a sexually transmitted disease. So that is a virus that causes cancer. I don't know if you guys remember this commercial, but there used to be a commercial about 10 years ago where for Gardasil, and the person said, "Cancer caused by a virus? I didn't know that." Well, at the end of this PowerPoint, we'll actually talk about how scientists have known that viruses can cause cancer for over 100 years. But anyway, I wanted to give you some facts on cervical cancer. Approximately 273,000 women die each year, and HPV is found in 99.7% of all cervical cancers. There are more than 100 strains of HPV, some of which cause things as benign as warts, but 13 cause cancer. Gardasil targets two strains of HPV that cause 70% of all cervical cancers. Those are strains 16 and 18. By the age of 50, 80% of women will be infected with HPV, and 90% of those cases will be cured and do no harm. But some of the cases will lead to chronic, long-lasting infections that can lead to cancer. And those are the cases which we're talking about here. Okay, so what are viruses? Viruses are obligate intracellular parasites. So what that means is that they can only really replicate themselves inside of a host cell. Okay, they're obligate. They require a host in order to replicate themselves. So they are not living things. And if you remember the three domains, domain bacteria, domain archaea, and domain eukarya, the viruses don't belong to any of those domains. Some people consider them more complex chemicals than living things and refer to them as nucleoproteins. So in the case of a naked virus that is lacking an envelope, a membrane bilayer on the outside, they would just be composed of nucleic acid covered by a protein coat. Okay, and their viral genome could be either DNA or RNA. Okay, and if you remember back to when I talked about all living things are cellular, they have DNA as their genetic material, and they contain ribosomes. Okay, and then those ribosomes are different between eukaryotes and prokaryotes. Here, in the case of viruses, they are not cellular. Okay, in the case of an RNA virus, they do not have DNA as their genomic material, and they don't contain ribosomes. Okay, because as parasites, they would infect their host and then hijack all of that host cell machinery to make their own nucleic acids and their own proteins to replicate themselves. So just going over some terminology here, the viral host range would be the spectrum of hosts that a virus can infect. And that could be a broad host range, or it could be a very, very narrow host range. In the case of West Nile virus, it actually has a broad host range, so it can infect birds, horses, humans, and mosquitoes. And so not all viruses have a broad host range like that, but West Nile virus does. With regard to viral specificity, now we're talking about the specific cells that a virus would infect. And the example that I give here is HIV, okay, the human immunodeficiency virus. It is only going to infect not just white blood cells, but one particular class of white blood cells, which would be the lymphocytes. And more specifically, the CD4 positive lymphocytes. And we'll talk more about those when we get to immunology. I just want to mention the figure here, these tulips, the striped tulips. That striping is actually caused by a virus. Okay, so here in this image, we see a little cartoon of actually an enveloped virus. So you can see that membrane bilayer outside of the nucleoprotein, okay, or the nucleocapsid. The protein coat is referred to as the capsid. So inside that protein coat, you would have the nucleic acid core. Okay, it could be DNA, RNA, it could be single-stranded or double-stranded, linear or circular, or segmented. And then you have the protein coat, okay? And the protein coat, the capsid, is actually composed of individual capsomeres. Okay, so the capsomeres would be globular proteins, but they would be assembled into this usually a polyhedral shape. Okay, and then that would be referred to as the capsid. We'll look at the different capsid shapes. Okay, so you can have a helical shape or you can have this polyhedral shape. We'll look at that on the next slide. If the complete infectious particle, okay, which is something that we would refer to as the virion, if the virion is a naked virus, it would just be the nucleic acid and the protein coat. But, okay, if the complete infectious particle is enveloped, if it contains an envelope, then you would have a membrane bilayer outside of that nucleocapsid. In this figure, you can see that the membrane bilayer has transmembrane spanning proteins embedded in it, okay, and they're glycoproteins, okay, so they're proteins with sugars attached, and those proteins are actually what's going to recognize the host cell, okay, so that's actually what's giving the virus its specificity, and that's how it would interact with a protein on its host cell and gain entry into that host cell and cause an infection. All right, so in this figure, we get an idea of the size of the virus. So we're only seeing like part of a human liver cell, and then we're seeing all of E. coli, okay, E. coli and all of the flagella all around it. It's polytrichous flagella. And we're only seeing part of the human liver cell because the human liver cell is approximately 20 to 100 times larger than a bacterial cell, than E. coli. And then you can see how much smaller the viruses are than E. coli. Okay, so when you think about, when you look at E. coli under the compound light microscope, how small it is, okay, these are even smaller and not really something that we can see with our compound light microscope. We would need to actually have an electron microscope in order to be able to see these viruses. Okay, so for the classification of viruses, like we mentioned, they're not part of the three domains. And so they are generally classified on their nucleic acid type or the structure of their nucleic acid. So we usually would talk about like the RNA viruses or the DNA viruses, and we could talk about the single-stranded RNA plus sense viruses. I'll explain what that means in a minute, or the double-stranded DNA viruses. And so that would be mentioning also the structure of their nucleic acids. The DNA or the RNA can be linear, it can be circular, it can be single-stranded or double-stranded. It can be segmented, so you can have multiple segments. Like, for instance, the flu is a segmented virus. When we talk about the different families, we use the suffix viridae. And so I'll in the next slides, I'll talk about some different virus families. And then when we're mentioning a particular genus of a virus, that the suffix would be virus. So an example of the classification for the herpes virus would be family Herpes viridae, right? So the suffix viridae, genus simplex virus, okay, so the suffix virus, and then the common name would be the human herpes virus 2. So like I mentioned, we usually classify viruses by their nucleic acid type and their chromosomal arrangements. Okay, and so for these classifications, the plus sense RNA virus or the minus sense RNA virus, what we're talking about with the plus sense, if you remember, like in DNA, the sense strand, right? The sense strand would be transcribed, and that would be what would be coding for the protein. The plus sense RNA is actually that RNA, that genomic RNA of the virus, is equivalent to an mRNA. And so when that virus infects its host, ribosomes can actually bind to the genomic RNA and be translated into protein. On the other hand, the minus sense RNA viruses, the minus sense is complementary to the plus sense. And so in that case, you would need to make the plus sense from the minus sense template. All right, and so you'd need to make an RNA copy of an RNA to get the plus sense RNA, which would be the mRNA, which could then be translated. All right, so if you think about, the whole flow of genetic information from gene to protein, we replicate our DNA, we transcribe it, RNA polymerase transcribes an RNA from a DNA template, and then we can make protein from an mRNA, but we don't really have an enzyme that can make RNA from an RNA template. Okay, but that's something that viruses do, and they do have those RNA, they're called RNA-dependent RNA polymerases, but for them to be able to infect the host and make that plus sense mRNA equivalent, they would need to carry that enzyme in the particle, okay, in the infectious particle, the virion. All right, and we'll talk more about that when we look at the particular infection life cycles of the different viruses. There are also double-stranded RNA viruses, and they can be segmented. Like I said, they could be linear or circular. All right, so just to get into the different families of viruses, the first one that we're going to talk about is the Picornaviridae. So the Picornaviridae is the typical plus sense RNA virus. So it does not need to carry that RNA-dependent RNA polymerase in its capsid, because when it infects a host, it can immediately be translated to produce that protein, and then it can make the minus sense RNA, which can then be used as a template to make more of the plus sense RNA. You need more of the plus sense RNA because that's the genomic RNA, so you need to be able to replicate it, right? All right, and we'll talk more about that when we look at those specific slides. But in the figure, in this electron micrograph, you can actually see that the virus is like a little naked virus. Okay, you can see the polyhedral capsid. You can see that it is very, very small. About 18 to 30 nanometers in size. Right, if you remember when we were looking at that slide that had the sizes on it, a ribosome is about 25 to 30 nanometers in diameter, so this is even smaller than a ribosome. And its name, the picornavirus, actually, if you break that down, pico-RNA virus. Okay, so it's an RNA virus, and pico, if you remember the prefixes for the metric system, there's like micro, and then nano is even smaller than that, and then pico is even smaller than that. So this is pico-RNA virus, very, very small, and then they just put that all together and they call it picornavirus. So an example of a picornavirus is the poliovirus. Okay, poliovirus is actually a picornavirus. All right, Retroviridae, okay, and HIV, the human immunodeficiency virus, is actually a retrovirus. Is also a plus sense RNA virus, but it is not the typical plus sense RNA virus like the Picornaviridae, because these viruses, okay, when they infect their host cell, they don't replicate themselves and lyse the host. They actually make a DNA copy of their RNA genome, and then that DNA copy gets incorporated into the host cell's genome as a provirus. Okay, I just want to mention that, you know, when we were talking about the phage, okay, the bacteriophage, and we were saying that the bacteriophage can incorporate themselves into the bacterial genome, and then they can be excised, right? With this type of virus incorporating itself into the host genome, it is never excised, okay? So the provirus would be in the host genome forever. Okay, so this is an enveloped virus, all right? You can see that in the transmission electron micrograph. Okay, remember the transmission electron micrograph, those are the cross sections, so we can see a cross section. We can see the membrane bilayer around the nucleocapsid. These are actually kind of big, right, as far as viruses are concerned, 100 nanometers in size. And the virion would contain 2 copies of the single-stranded plus sense RNA, as well as 2 copies of the enzyme reverse transcriptase. And reverse transcriptase is the enzyme that's going to make the DNA copy from the RNA. Okay, so, because remember, you know, that's not something that we do, right? Okay, so you know, if we're infected by HIV, the virus is not going to hijack our reverse transcriptase because we don't have that, right? So it's got to carry that enzyme within the virion, right? So that it can make a DNA copy of its RNA, and then that DNA copy would get incorporated into our DNA, okay, as a provirus. All right, and we'll talk more about the specific life cycle of HIV in the upcoming slides. This is a typical minus sense RNA virus, okay, Rhabdoviridae. It's a bullet-shaped helical virus, okay, so the capsomeres, instead of being arranged into a polyhedral head, they are arranged into this helix, okay, which results in this bullet-shaped capsid. All right, they are enveloped. All right, you can kind of see the hazy envelope in the figure to the right. They're large in size. And now, since this is a minus sense RNA virus, okay, it would need to infect the host and then make the plus sense, right? Because the plus sense is the mRNA in order to be transcribed. All right, so since it needs to make that RNA copy of its RNA genome, first thing, straight off the bat, it needs to carry that enzyme in its virion. Okay, it needs to carry that RNA-dependent RNA polymerase so that it can make that plus sense RNA copy from its minus sense genome. Okay, and then that plus sense RNA would be translated to make more capsomere proteins, okay, which it's going to need, right, in order to replicate itself. All right, so here's an example of a DNA virus family, all right, herpes viridae. Okay, you can see that herpes viridae is an enveloped virus, right? The envelope is actually pretty fragile here, and herpes viridae, you know, a lot of people used to think that, you know, you could pick it up off of a toilet seat or something like that. And I'm not saying that that could never happen, but I don't think that that's a super common form of transmission. Because the envelope is so fragile and the envelope needs to be intact in order for the virus to be able to cause an infection, it's not a super common way that that this virus would be transmitted. So the herpes virus is a double-stranded DNA virus. It's a linear DNA virus. It's enveloped. You can see that in the transmission electron micrograph. It's a little bit larger in size, 120 to 200 nanometers, and its double-stranded DNA would be incorporated into the host chromosome, okay, as a provirus. It causes what's referred to as a latent infection. So it can go for a long time without being replicated, and then it can replicate. It can be induced to replicate. So it can live in nerve cells, but then at the time of activation, like say if you had a fever or a sunburn, it can be activated, and then you would show symptoms. Okay, you would show the blisters, right? Only 10 to 15% of people who carry the virus actually show symptoms. Okay, so this is an example of a DNA family of viruses. Well, I wanted to talk about just some causes of emerging viruses. Of course, we have a situation with an emerging virus right now, the coronavirus. Um, uh, and the examples that I'm going to give you here of emerging viruses don't really address the coronavirus, but the first example of how a virus could become an emerging virus, okay, so how you could see like an increase in viral disease would be when you have a virus that is previously endemic, okay, which means that it would cause a low level of, constantly is causing a low level of infection in a certain area. Like, for example, dengue in the Caribbean. Dengue is always causing a low level of infection in the Caribbean. But then, all right, due to, some type of factor, you can have an increase in viral disease, okay? And as far as dengue is concerned, it started to move north as global warming progressed. There were cases of dengue actually in Florida in 2010. Dengue is carried by Aedes aegypti. So is the next viral disease that I'm going to talk about, which is yellow fever. So Aedes aegypti is a mosquito. And they never thought that mosquito would be able to make it, kind of like Zika. They never thought that mosquito would be able to make it to the U.S. and then that there would be person-to-person transfer, like from a mosquito biting an infected person and then going and biting someone else. But there actually were cases of Zika and also cases of dengue in Florida that were caused this way. Also, okay, another way that you could have an emerging viral disease would be through coming into contact more frequently with the viral vector, okay, in this case, the insect. And there were cases of people going further and further into the tropical rainforest, clearing it to use it for farming, and there was an increase in the cases of yellow fever. Yellow fever causes flu-like symptoms. There's liver damage, which causes yellowing of the skin. It's normally endemic in Central America and tropical South America, but you can actually have an increase, a spike in the viral disease due to this kind of invasive movement into once tropical areas in order to farm them. So the flu is actually always considered an emerging disease, and the reason why is depicted in this figure right here. So the flu actually has a broad host range, and so the flu can infect birds, it can infect humans, it can infect pigs, right? Swine flu. And if you have a host, okay, and that's what they're depicting in this figure right here, that's infected by two strains of the virus, okay, so like it's being infected by the purple virus and the orange virus, once that those two viruses are replicating themselves, they could actually swap parts of their genome. And so that's what you're seeing with the viral particle that has some purple and some orange, and create a new mutant type virus that basically would not be, that our immune systems would not be able to recognize. So say, for instance, that the new strain of the human flu virus had a flu virus core, but it was covered by a pig type capsid. Okay, that would be something that our immune systems would not be able to recognize. And if this happened, okay, there would be a worldwide pandemic like what we're going through right now, where no one would have immunity, and basically we wouldn't be able to make a vaccine in time, okay, because usually you need about 9 months to develop the flu vaccine. The flu is actually highly studied by the CDC and the World Health Organization. They study all of the different strains of the flu that are occurring worldwide, and then they grow those viruses in eggs, actually, in order to make the vaccine. And so, every year the flu vaccine is a little bit different depending on what strains were out there. They're trying to basically, guess, or predict what recombinations might occur, and give us a vaccine that would protect us against these recombinations just in case they occurred. If we had, some type of immunity built up to these proteins that would be on the outside of the flu virus, we could, have some type of immunity that could be able to fight the flu. But okay, if we don't know what's out there, okay, and we don't create the appropriate vaccine, then, we could have some type of flu pandemic. The epidemiologists, and I'm sure you guys have already heard this, recently, but they say it's a matter of when and not if that there will be a worldwide flu pandemic. We really need to have a universal flu vaccine, but that is another thing that's like kind of a long time in the making, because for us to be able to create a vaccine that has, all of the H parts and all of the N parts, right? Like H1N1, H3N1. We need to have all of those H parts and all of those N parts in the vaccine. It hasn't happened yet, and I think there are some challenges with trying to do that. I have down there the swine flu pandemic of 1918. I'm sure you guys have heard about this, but it killed between 50 and 100 million people in 1918. What I've just heard recently, which was so interesting, was that they said that after World War I was finished, that a bunch of people were like kind of self-isolating themselves and maintaining the social distance. They kind of ran out into the streets to celebrate, and after that happened, there was a huge spike in the swine flu. There was another spike. So they showed that social distancing helped tamp down the number of cases of the swine flu in 1918. And then they also showed that, kind of with the end of the World War I and people celebrating how like lifting those restrictions could lead to another spike in the number of cases. Okay, so we're going to go through the replication cycle for the plus sense and the minus sense viruses, and not only the animal viruses, but also the bacteriophage. And so there are these 5 steps, okay? Adsorption would be the attachment of the virus to the host cell. Then penetration would be how the virus gains entry into the host cell. Synthesis would be where the virus is hijacking all of the host cell's machinery to replicate its proteins and its nucleic acid genome. And then maturation would be the spontaneous assembly of all of those parts. And release would be how the virus leaves the host cell. Okay, there's a couple of different ways that can happen. Okay, so we'll go through this replication cycle, those 5 steps for animal viruses, as well as the bacteriophage, okay, the virus that infect bacteria. Okay, well, here's a little cartoon and a transmission electron micrograph of a bacteriophage. These are the T-even phage, okay, so like T4, T2, T6. They infect different strains of E. coli. They are double-stranded DNA phage, and they have this complex capsid. So they actually have a more complex capsid than most of the animal viruses. Okay, so we saw with the animal viruses that they could have a like an elongated polyhedral type capsid, but here the bacteriophage have a polyhedral capsid, they have a helical sheath, and then they have these tail fibers. Okay, it's the tail fibers that actually attach to protein receptors on the surface of the cell. And so it's the tail fibers that really account for that specific, for the specificity basically of the phage for its host. Okay, so I mentioned that for these T-even phage, the T2, T4, T6, that each one of them binds to a different strain of E. coli. So there would be differences in those tail fibers, which are binding to different receptors on the host cell. Okay, and that would be how the phage would gain entry into the cell. The T-even phage are virulent phage. They cause the lytic cycle. Okay, you might remember from chapter 8 when we talked about transduction. We talked about how you could have virulent phage, which enter into the lytic cycle, and then also temperate phage, which enter into the lysogenic life cycle. These phage would cause the lytic cycle. So they would lyse the host cells, and then they would go off and infect neighboring host cells and go through the whole life cycle again, okay, where then they would lyse those host cells, et cetera, et cetera. Okay. All right, so this is just a little summary slide that talks about the different parts of the bacteriophage. So with the phage, the genomic DNA from the head, okay, which is that polyhedral head, that's where the genomic DNA would be. When you have an infection, the DNA is going to move from that head through the sheath, and the sheath is actually going to retract to kind of inject the host cell genome into, the bacteriophage genome into the host cell. The tail fibers are binding, they're making that specific protein-protein interaction with receptors on the host cell. Okay, so this slide looks at the lytic cycle of the virulent bacteriophage. Okay, so in the first slide, you can see, in the first part of this slide, number 1, adsorption, you can see where the phage is binding to the bacterium. Okay, and so it's like the tail fibers are making specific protein-protein interactions with receptors on the surface of the bacterial cell. Okay, and then in the second step, penetration, you can see where the phage, okay, the sheath is retracting, and the DNA is being injected from the phage head into the bacterium. Okay, and the whole protein coat of the phage is left on the surface of the bacterium. All right, we're going to see when we talk about animal viruses that this is different from an animal virus infecting its host cell. So with an animal virus, the whole nucleocapsid would enter into the host cell, and then part of penetration with the animal virus would be uncoating, where you've got enzymes that would enzymatically remove that protein coat. Okay, but here for the bacteriophage, the protein coat is left on the surface of the cell, and it's just the nucleic acid that would be injected into the host cell. Okay, so then after that happens, the bacterial chromosome is broken into pieces, and the phage would hijack the host cell's machinery and replicate its DNA and make its own capsid proteins. And so this stage would be called biosynthesis. And then during maturation, you would have the phage particles spontaneously assembling. Interestingly, you can actually take all of these different phage parts and put them into test tubes, and they will spontaneously assemble. Okay, and so this is basically what's happening during maturation. And then with the lytic cycle, okay, you would have lysis of the host cell, and all of those phage particles would be released, and they would go off and infect neighboring cells, and the whole cycle would start all over again. Okay, so these are just some notes on what I just said. With adsorption, okay, that's the binding of the bacteriophage to the host cell. You would have that specific protein-protein interaction. And then, okay, I probably forgot to mention that the bacteriophage would have the enzyme lysozyme. All right, remember lysozyme is an enzyme that would break the peptidoglycan between the NAMs and the NAGs. It would enzymatically digest the peptidoglycan. Okay, so this would kind of weaken the cell wall of the host cell so that the virus could inject, it would kind of create a hole so that the virus could inject its DNA into the host cell. Okay, and then synthesis would occur. All of the viral parts would be made. In maturation, all of those viral parts would be assembled, and then release. Okay, as far as the lytic cycle is concerned, you would have the rupturing of the host cell, okay, basically the killing of the host cell, and all of those new virus particles would go off and infect neighboring cells. Okay, so we can actually create a bacteriophage growth curve, just like we created a bacterial growth curve, but it's a little bit different. Okay, in the so in the figure, you can see the total virus is the green line, and then extracellular virus. Okay, so this would be after the host cell was lysed, okay, and the virus were released, okay, that's indicated by the blue line. Okay, so the eclipse period, all right, would be the time from penetration, okay, that would be when the DNA would be injected into the host cell through synthesis. Okay, so when, all right, the DNA is broken down, the host chromosome is broken down, and then the bacteriophage start to make their own proteins and replicate their own DNA. The latent period goes from the time of penetration all the way to the time of phage release. Okay, so you would have penetration, the DNA would enter into the cell. Okay, the latent period would also incorporate synthesis, okay, maturation, all right, and then release. Okay, so you can see that that's going from where the total virus is now the extracellular virus. Okay, so it becomes the extracellular virus because you have release, the lysis of that host cell, and you have release of those viral particles out. Okay, and now they're infecting neighboring cells. The viral yield could also be called the titer, and that would be the number of viruses that would be released per infected cell. Okay, so like the figure shows, it's usually about 200 per host cell. So you have one virus infecting that host cell and replicating itself, making 200 particles. Okay, and then when that host cell lyses, those 200 virus would go off and infect neighboring cells. So when this happens, okay, like on a lawn of bacteria, you have virus, you have bacteriophage lysing cells, you would actually create a clear area where the phage has infected the host, and that host is lysed, and then those phage have gone off and infected the neighboring cells and lysed them. Okay, you would create this clear area, which is referred to as a plaque. And so this is a little plaque assay where the number of phage was diluted. Okay, so what does this kind of remind you of? It kind of reminds you of the standard plate count, right? But instead of diluting the bacteria, we're diluting the phage. Okay, the amount of bacteria that we add to create the lawn is going to be absolutely the same for every dilution of phage that we have, but we're going to dilute the phage and we're going to try to get a countable plate. Okay, so in some situations we would have the number of phage would be too numerous to count. Okay, the plate might just look clear because everything's lysed, all of the bacteria are lysed. And then, so that would be too numerous to count. And then on some of the plates, you might only have one plaque, okay, which would be too few to count. And then you're going to find that plate that has between 30 and 300 plaques. And basically, to get the number of phage per mil, you would take that plaque count and you would multiply it times the dilution factor. So if you diluted something 1,000-fold and you got your plaque count from that plate, okay, like say you got 50 plaques, okay, you would multiply that times 1,000 to get the number of phage per mil in the original stock culture. Okay, and then we would report these plaques that we see not as the number of phage, but as plaque forming units. Okay, so it's kind of like colony forming units, right? We didn't say when we saw a colony of bacteria that that was from one bacterium, okay, because it could have been from an arrangement, right? So we're calling these plaque forming units and not bacteriophage per mil. Okay, so we would say like PFUs per mil. All right, so with lysogeny, the bacteriophage doesn't immediately kill the host cell. Okay, we'll see actually on the life cycle for temperate bacteriophage that there is a point, this induction point, where the phage can cause the cell to enter into the lytic cycle. Okay, but a temperate phage would not immediately enter into the lytic cycle. It would cause the cell to go into this lysogenic cycle. So when you have the bacteriophage incorporating itself into the host cell's chromosome, that's referred to as a prophage, okay? And with the bacteriophage, that can be excised. Okay, so when you have a bacterium with the temperate phage incorporated into it, that together is referred to as a lysogen. You guys might find when you do your disease presentations that a lot of the disease agents are actually lysogens. So they've got this, prophage, and it's the prophage that makes the particular toxin that makes them virulent. So you might find that when you're doing your disease presentation. So lysogenic conversion results when you make a couple of proteins, okay, one that would stop the virus from replicating itself, okay, and then the second one, which would prevent the same phage from infecting that cell. Okay, so that would, once those proteins are made, okay, you would have this complete lysogenic conversion where the cell has this prophage incorporated into the bacterial chromosome, okay, but you're not entering into the lytic cycle because the phage is preventing itself from replicating, and you're also preventing the cell from being infected by that same type of bacteriophage. All right. But at some point, okay, you can have induction. It can be induced or it can be spontaneous, and the prophage would be excised, and then that particular cell would enter into the lytic cycle. Okay, let's take a look at the figure. All right, here's the figure. Okay, the lysogenic cycle is at the bottom. All right, so we would start over here with 1. All right, 1 and 2, absorption and penetration. Okay, so the phage, now this is a temperate phage, would bind to the bacterium, inject its DNA. Okay, the DNA would be incorporated into the host cell chromosome as a prophage, and for as long as that cell divides, okay, it would and so here it shows indefinite cell cycles, right? It would just replicate that phage DNA along with its own DNA, okay? But then at some point you can have induction, right? And the prophage would be excised from the bacterial chromosome. The bacterial chromosome would be broken into pieces, and then you would go through the lytic cycle, okay, where you would have biosynthesis, maturation, and release, okay, and the release would actually lyse the cell. We saw in the previous figure, okay, so it would be an enveloped virus. The viral glycoproteins would bind to receptors on the surface of the host cell, okay, and then you would have those two membranes, okay, the envelope and the cell membrane of the host fused together, okay, and the nucleocapsid would gain entry. Okay, so with HIV, right, it's actually an enveloped virus, okay, so you would have the viral glycoproteins binding to receptors on the surface of the cell, the two membranes fuse, okay, you have penetration, uncoating, the enzymatic digestion of the protein coat, and then you have this plus sense RNA virus, okay, but it's not going to behave like a regular plus sense RNA virus. It's actually going to use, okay, so inside that protein coat, it also has a copy of the enzyme reverse transcriptase, and it's going to use that reverse transcriptase to make a cDNA, okay, C for copy, cDNA copy of its RNA genome. Okay, so you would first make the DNA, okay, from the RNA template, and you'd have this DNA RNA hybrid, okay, and then the RNA would be digested away. You would use the host cell's replication machinery to make double-stranded DNA, and then that double-stranded DNA would be incorporated into the host cell's chromosome, okay, as a provirus. Okay, and then, all right, as DNA, it can just be transcribed. Okay, the transcription would make the plus sense RNA viral genome, and then that can be translated to make reverse transcriptase and to also make the capsid protein. Okay, you can have assembly, all right, and so you assemble 2 copies of the plus sense RNA and 2 copies of the reverse transcriptase into a capsid, the protein coat, okay, and then that capsid would bud from the surface of the cell, okay, and the cell membrane would become the envelope of HIV, okay, and you would have the viral glycoprotein spikes would be expressed on the surface of the host cell and be incorporated into that envelope. Okay, so here are the notes on that, just so that you know. And then where does the envelope come from? Okay, in budding. Okay, I just mentioned it, right? So the envelope would be from the host cell. Okay, the host cell's plasma membrane. Okay, that would be where the virus would get its envelope, all right, when it buds from the surface of the cell. Okay, now that's not going to kill the cell immediately, all right? That doesn't kill the cell outright like lysis would, okay? But eventually it will kill the cell, okay? And that's what accounts for that low lymphocyte count with HIV. Okay, so I'm going to skip this. All right. So I just wanted to talk a little bit about culturing animal viruses. I mentioned when we were talking about the flu as an emerging disease that, to make the flu vaccine that you would need to, grow the virus in eggs, right? So this is actually still a really good host system for the flu virus, influenza A. And especially I think if you get the nasal vaccine, they ask you if you're allergic to eggs. They might ask you that for the intramuscular as well. And the reason why is because the flu virus is grown in eggs. Okay, so intact fertilized eggs are used to grow animal viruses. But if you're studying the effects that a virus has on cells, okay, the cytopathic effects, you can't really see it inside of an egg because of the shell. Okay, so to culture animal viruses, you can actually use eukaryotic cells, and there's a bunch of different types of cell cultures. But just in general, animal cells are grown in a very different way from bacteria. Okay, so bacteria, we've seen how you can grow bacteria in suspension, right? Okay, so they can grow in a liquid suspension. Animal cells need to grow attached to a surface. So they grow in monolayers, okay, attached to a surface in culture. And they'll grow and divide, okay, until they take up all of the space on that surface and they become kind of crowded. And then what you can do is you can subculture them. So you can take just a fraction of them and reseed a new dish. Okay. And then, they'll have more room to grow and divide and fill up the entire bottom of that new dish. So because they make these adhesions to the plastic dish, you actually have to treat them with proteolytic enzymes. Proteolytic enzymes are enzymes that cut proteins. So they would cut those protein attachments, and then you could suspend them in solution and just take like maybe 1/10 and reseed a new dish. And this is what we would call subculturing. So you can subculture the culture, a certain number of times. Primary cell culture refers to cells that come directly from an animal. You can, take cells from an animal and usually by giving them the right growth factors, you can kind of trick them into dividing a few times. So that would be primary cell culture. There are diploid fibroblast strains. I'll tell you why diploid is important in just a minute. But these diploid fibroblast strains come from fetal tissue. So remember, when you have fertilization and the cell divides and you create a fetus, all right, for that fetus to grow to the adult size, it's going to go through a lot of mitotic cell divisions, right? So these, immature cells that are coming from fetal tissue, they have retained the capacity for repeated cell division. So you can grow them in culture and passage them a bunch of times, like maybe 60 times. They can divide. So you could grow those in culture for quite some time. There are these continuous cell lines, and these are cells that are not necessarily diploid. They're not necessarily normal cells, and they will just grow and grow and grow. And an example of those, of a continuous cell line, would be the HeLa cell line. I don't know if any of you have heard of the book, The Immortal Life of Henrietta Lacks, but they're referring to this cell line being immortal. So they've been growing this cell line in culture since 1953, and it just completely, keeps dividing. It's just gone through an infinite number of divisions and shows no sign of slowing down. This cell line, the HeLa cell line, came from Henrietta Lacks. She was a woman who had cervical cancer. And at that time in the 50s, they could take cells from people and grow them in culture without their permission. And it turns out that her cells were one of just very few cells that had this ability to be immortal. And it had to do with a lot of different factors. But let me just say that these cells are not normal cells. Oftentimes, when you have a continuous cell line like this, you can go through replication without going through cell division. So now you don't have a diploid cell, you have a polyploid cell. All right, so these are really abnormal cells, and you wouldn't want to study, like if your lab was studying the cell cycle or something like that, you wouldn't want to use these cells because these cells are really messed up. But if you wanted a cell that you could express a protein in, you could definitely do it in this cell line. All right. So those are the different types of cell culture cells that you could use. Now, the reason why we would want to, have eukaryotic cells instead of eggs would be to look at the cytopathic effects that a virus has on a cell. And here in this figure, you can see the cytopathic effects that these would be the visible effects that a virus infection has on cells. So on the left-hand side, those cells that are all spread out, okay, and they're making these adhesions to the tissue culture plastic, okay, that's what normal fibroblasts look like. But when they're infected with a virus, they detach from the culture, okay, and so they've broken those focal adhesions and they're kind of rounded up, okay, they're kind of balled up and they're detached from the culture. Okay, so that would be one of the cytopathic effects that a virus could have on a culture, on cells, and we would never be able to see that in an egg, right? Because the egg has the shell. Okay, so but we can see that in tissue culture. Okay, another cytopathic effect that viruses have on cells would be transformation. Okay, and transformation here, we're referring to the transformation of a normal cell to a malignant, metastatic cancer cell. Okay, so cancer can be caused by a virus, and cancer is the uncontrolled cell growth of abnormal cells. Oftentimes when these normal cells are transformed into malignant cells, you're activating a growth factor pathway, okay, which causes the cell to grow and divide. You're activating it, even in the absence of the growth factor, you're activating some proteins in that pathway that causes the cell to grow and divide. And that, maybe was a pathway that was turned on in early development, but turned off later in life. But now that pathway is being reactivated. And so you have this uncontrolled growth of these transformed cells. Okay, and that could lead to a tumor. Okay, so an accumulation of cells. And tumors can be either benign, which means that they don't spread from their original location, or they can be malignant. Okay, and that would be a tumor that is spreading from its original location, spreading or metastasizing. Okay, so it's moving from its original location. Benign tumors, they're not necessarily as benign as they sound. Like if you have a tumor in your brain, okay, that is not, a cakewalk, right? I mean, that's, it could be a benign tumor, but it's still a huge problem, right? But when you have a malignant tumor, okay, which is metastasizing or spreading throughout your body, that would be when a lot of people consider that you have cancer, right? Okay, when you have this spread of the transformed cells causing tumors all throughout your body. Okay, and all right, it turns out that 10% of cancers arise from 10 to 15% of cancers arise from viral infection. Okay, and I mentioned, at the beginning of this PowerPoint lecture that that commercial where the person was saying cancer caused by a virus. I didn't know that. Well, they've actually known that for more than 100 years. So in 1911, a farmer noticed that his chickens had these huge sarcomas, okay, which would be tumors in the connective tissue, and those tumors were studied by someone named Rouse, Rous, and what he found was that you could take a cell-free extract. Okay, so what's smaller than a cell, right? A virus. You could take that cell-free extract, the virus, and infect a new chicken. And so that virus is the Rouse sarcoma virus, RSV. Okay, and so that was discovered in 1911, okay, that you could have a virus that caused cancer, okay, caused this sarcoma. All right. So this is just, just looking at, it's a very simple figure looking at the human papillomavirus. And the human papillomavirus is a double-stranded DNA virus. And when this virus, okay, especially those couple of types that I talked about, right, 16 and 18, when they infect a cell, all right, they can exist as a provirus, and what they would cause that cell to do would be to have uncontrolled cell growth. Okay, so the cytopathic effect for this particular virus would be uncontrolled cell growth, and that would result in a tumor. Okay, so you also make these viral replication proteins, and the viral replication proteins block the effect of tumor suppressor genes, okay, which would normally suppress the growth, the uncontrolled cell growth. And so then as a result, you have uncontrolled cell growth and you have tumor formation. Just to talk just briefly about a couple of virus-like agents. Viroids are smaller than viruses. They're just RNA, okay, no protein coat. And for the most part, they mostly cause lethal diseases in plants, not really so much us. But what they do is, and there are no protein products involved, okay, what they do is they disrupt the host cell's metabolism. And so what they're thinking happens there is that the RNA maybe interacts with an mRNA and prevents it from being translated, okay, and that would be what would be causing the lethality there. Prions, I don't know if you guys have heard of prions. Mad cow disease is a prion disease. But this is another disease agent that in this case is protein only. Okay, so that's really odd as well, right? Like all of the all of the disease agents of humans, whether they're bacteria or viruses, they all have nucleic acid as their genetic material, even if it's not DNA. Okay, but here with prion disease, there is no nucleic acid. It's protein only. So we actually have the prion protein in us, okay, and that would be, what we see in part A of this figure. Okay, so that's the normal protein. And you can see that it has a lot of alpha helices in it, right? But when it comes into contact with the prion protein, okay, so when it touches a prion protein, it becomes this different shape, all right? And so you can see those arrows in the B part of this figure. Okay, that's beta pleated sheet. Okay, so it becomes this different shape, and it becomes a prion protein, and those prion proteins can stick together. Okay, and when they stick together, okay, they kill cells and they form plaques. Okay, in the brain. Okay, which creates this spongiform, spongy brain encephalopathy. Okay, so BSE, bovine spongiform encephalopathy, mad cow disease, is this spongy brain disease. Okay, but this disease, like, it can actually occur in other species. There is a prion disease that affects humans. It's called Creutzfeldt-Jakob disease. It's a spongiform encephalopathy. And it usually affects people later in life. And what they think that is due to is they've done studies on these prions, and you need to have a certain number of them before they cause disease. So there's like a threshold number that you need before they cause disease. And that is usually associated with, someone living like their entire life, right? Through the course of their life, they've accumulated, enough prion proteins to where they actually have the prion disease. But there are some new variants that have actually infected younger people as well. Okay, I just heard, not too long ago that they were saying that they feel like all forms of dementia are caused by prions, but actually there's going to need to be a lot more studies done on that before they can say that conclusively.